So it's my pleasure to be here today, and I'd like to thank the Linnean Society for giving me the opportunity to talk to you guys about the science that I find interesting. So my name is Ravinder Kanda, and I'm a researcher at Oxford University, and broadly what I'm interested in is genome evolution. So how do genomes get to be the way that they are today? What are the processes that shape our genomes and have shaped them in the past? So a quick introduction. I'm sure that most of you know this stuff already. What is it that we mean by the genome? Well, essentially, we're talking about the genetic material of an organism. We're talking about all of the chromosomes that are present in the nucleus of the cell. And if we look a little closer at these chromosomes, what we find is that they are tightly wound up strands of DNA that, when uncoiled, reveal the double helix structure that I'm sure that a lot of you are familiar with. The double helix structure consists of a structural backbone and the four building blocks, the nucleotides of DNA. Now, some of these stretches of DNA will code for genes. And in 2003, the sequencing of the Human Genome Project revealed that the human genome was a staggering 3.2 billion base pairs long. But out of this, only 20,000 genes are approximately present, which makes up a very small proportion of the genome. So this is represented in the pie chart here, and this green sliver down here shows that only 2% of the genome is actually protein-coding genes. The rest of the stuff was termed junk DNA and split into several groups. We can tell that these are pieces of DNA that are related to each other, which is how we can classify them into separate groups. And it's one of these groups in particular, this group here, the LTR Retrotransposons, that I am interested in currently. So these LTR retrotransposons are actually the remnants of viruses. So if we look at this chart here, which shows us all the viruses that we know about today, and they can all be classified into these various groups, depending on whether their genetic material is RNA or DNA, and you can see that they vary quite considerably in size, in structure, and their genome size, which is given down the bottom, also varies quite considerably here. Now, one particular group of these viruses is called the retroviruses, and that's these guys here. And this is what I'm going to be talking about mainly today. So what does a retrovirus look like? So generally, a retrovirus has a structure, a bit like this. We have an envelope around it, and it has glycoproteins on its surface. In the center, it has a capsid, which contains 2 RNA molecules and the reverse transcriptase enzyme that it needs. Now, when we look through the genome, what we actually see in terms of retroviruses is a structure that looks a little bit like this. So the black bits represent the DNA of the host organism, and we have 3 genes in the middle, GAG, POL, and ENV. And these 3 genes encode everything that the virus needs to make copies of itself. The GAG codes for the core structural proteins, the POL codes for the polymerase, and the ENV gene is the envelope gene that allows it to gain an envelope as it leaves the cell. Now, these things get into the genome, and then they sit there until they're inactivated by either mutations, which occur in the genome, or by recombination occurring between these 2 LTRs, which takes out this middle region and leaves us with something that looks like this, a solo LTR. So even though we might not have the full virus in the genome, we can still tell that at some point in time, one was in the genome. So how do we get from our viral particle, which looks like this, to something that looks like this in the genome? Well, this particular group of viruses has to replicate itself by integrating into the genome. So the viral particle approaches the cell, and the envelope of the viral particle allows it to fuse with the cell membrane, allowing entry into the cell. the capsid is released, and the RNA is reverse transcribed into DNA, which is then transported into the nucleus of the cell. Once it's in the nucleus of the cell, it can use the cell machinery to make more copies of its viral genomic DNA. It gets translated and eventually ends up reassembling and leaving the cell and going on to infect other cells. Now, on occasion, these viruses manage to integrate into the sperm or egg cells of an organism. And when they do this, they are then inherited by all of the offspring of that particular individual. At this point, we call them endogenous retroviruses. So, we're not unique in this respect. Endogenous retroviruses are found in the genomes of all mammals, reptiles, the other primates, the big cats, and even the more distantly related mammals, like even the more distantly related vertebrates, like the lamprey, for example. And across all of these species, endogenous retroviruses account for a considerable amount of their DNA. In mice, the figure is thought to be about 10 to 12%. In humans and primates, it's about 8%, which is a considerable amount when you consider that only 2% of our genome contains genes. Now, this 10%, it didn't all happen at once. It would have been many retroviral infections that have happened over evolutionary time. Now, when you consider that the human body carries out roughly around 10 billion cell divisions in a lifetime, the replication and maintenance of all of this viral junk requires a considerable amount of energy. So why do we keep it around? So before I get on to why we do that, I'd just like to give you an overview on how studying retroviruses can be informative in many ways. So from the endogenous ones in the genome, we can gain some evolutionary insight into what has happened in the past of that particular host. So it tells us something about the viruses that have bothered that particular species across all time, like a snapshot in the past. The genome of each organism is like a viral graveyard in this respect and gives us some idea of how long a particular virus or a particular family of viruses have been infecting that species. Viruses have also had a considerable impact on the genome evolution, and I'll talk more about that a little later. Now, for some of these viruses, there are also medical implications, and it also gives us some insight into how we have dealt with these viral infections in the past. And then there are some more practical uses of the viruses that we can use today. So one of the reasons that we possibly keep all of this viral junk DNA around is the option of gene co-option. So what do I mean by that? There are numerous examples in the genome where we have stolen a virus gene and used it for our own benefit. One of the most famous examples of this is a gene called syncytin, which is essential in the formation of the placenta. So after 5 to 6 days, when you have your fertilized egg cell, the fertilized egg cell has to attach itself to the lining of the uterus. And it does this by embedding itself in the side, and then these cells here, which make up the trophoblast, express syncytin. The syncytin makes these cells fuse, and this essentially forms the placenta. Now, cell-to-cell fusion is not really a feature that is quite common in mammalian cells, but it is something that we do find quite commonly in viruses. Viruses use this cell-to-cell fusion when they enter the cell. Their envelope fuses with the cell membrane, and in this sense, that gene is perfect for this function. So when we consider all the placental mammals, which originated about 150 million years ago, they would have acquired the envelope gene of a specific virus, which allows us to replicate or reproduce as we do today. So this here on the right is a tree that shows how different groups of mammals are related to each other. So it's kind of a similar concept to a family tree, which I'm sure that most of you are familiar with. Just as in the same way with a family tree, you can see how you are related to your siblings, to your parents, and to your ancestors. We can display this information about the relationships between various groups of mammals by looking at their DNA sequences and figuring out which ones are more similar than others. If we know the rate at how the genome changes, what we can do is put a timeline on this. So this is what we see today, the present time, and working back, we can see where one specific lineage would have split into two to form the different groups of mammals. So we know that with placental mammals, they must have acquired this envelope gene once in the past. But when we actually look at the structure of the placenta, it looks very, very different in different groups of mammals. So why might this be the case? By comparing the syncytin gene in all of these mammals, we can see that some of these syncytin genes that are present are more closely related to viruses than they are to each other, which indicates that it's not the version that was inherited or acquired at this point in time. The syncytin gene in humans, for example, which was acquired here about 20 million years ago, roughly, is actually very closely related to an endogenous retrovirus called HERV-W. But the one that's present in mice, wherever the rodents are, there, is more closely related to a totally different retrovirus. So this tells us that subsequent to the initial acquisition of an envelope gene, we've also acquired one along the way that does a better job. And so it was co-opted into the genome, and it serves that function now. So this didn't just happen once. The acquisition of the envelope gene has happened 5 times in mammalian evolution, which is pretty impressive. So that's one of the reasons why we might keep around these viruses, so that we can steal their genes, put them to good use. It means that we don't have to develop our own. To develop a gene that serves this function would take millions and millions of years of evolution. In this case, we've acquired one from a virus. It's a little bit like an evolutionary shortcut. Another reason that we might possibly keep these genes around is to do with immunity. So with the number of whole genome sequences that we are getting from various organisms these days, what we are finding is that in some instances, having a copy of a retrovirus in your genome might actually provide you with an immunity benefit against infection by related external viruses. So one example exists in the sheep. In the sheep population, there is a virus called Yaxicte retrovirus, JSRV for short, which is not present in all sheep. In those sheep that don't have the retrovirus, when they encounter the external JSRV, they develop a usually fatal infection. It affects the lungs, and they die. But for sheep that actually have a copy of the virus in their genome, they seem to deal better with the retroviral infection that they catch if they encounter the external JSRV. And the reason for this is that the copy that is present in the genome actually produces particles that block the cell receptors. It means that the external virus can't get into the cell. It can't cause infection. Now, that's just one example. There are a number of other examples that are highlighted in this table, and all of them involve interfering with the retroviral life cycle. Some of the most well-studied examples of this are in cats with feline leukemia virus. There's an example in chickens with avian leukosis virus, and with mice we have numerous examples as well. The FV4 gene, for example, was acquired from the murine leukemia virus, and it seems to have been co-opted into the mouse genome to perform this very function of immunity. So that's one of the other reasons why we keep them around. Now, active endogenous retroviruses, or ERVs, we don't have any active ERVs in our genome. All of the copies that are present in our genome have been inactivated by mutation or solo LTR formation. But active copies do exist in the genomes of other organisms, as I've mentioned previously. And we can see that they are quite similar to external viruses that are not in the genome. In this way, we know that it's external viruses that are getting into the genome that make up this 8% in us. That must have happened many, many years ago. Now, if we have viruses getting into the genome, and they're kind of similar to the ones that are not in the genome, the external ones, we also have to be concerned about the possibility of new viruses arising. In cats, for example, it has been documented that you can get recombinant viruses, which are a mixture of the ones that are in the genome and ones that are not in the genome. And these then arise as viruses that go on to infect other cats. Additionally, we also have to be concerned about the risk of cross-species transmission, viruses from one organism infecting another. And we do actually see this happening today. There is an active retrovirus in the koala population, the koala retrovirus, which has only recently been discovered. It is decimating the koala population in Australia, but they have also found that it's actually integrating into the genome as well. Now, when we look at the DNA sequence of this virus, it's very similar to another virus that we find in gibbons, gibbon ape leukemia virus. So it's quite clear that koala retrovirus has originated from a jump from the gibbon ape leukemia virus. So on the subject of cross-species transmission, I'm sure that you've all heard of HIV, which is probably one of the most famous retroviruses that most people know about. So HIV, or rather the simian version that occurs in primates, SIV, is generally tolerated quite well by these old world monkeys. They have the infection, but they don't seem to get the same immune response that occurs in chimps and occurs in us. They seem to tolerate the infection instead. It doesn't kill them in the same way. Now, we know that HIV jumped into chimpanzees who do get infected and who do suffer with the infection as well. And then from chimps, it jumped into us. And obviously, HIV is a huge threat today, which is why we're interested in understanding it better. How did it make this jump? Why is it so lethal in this respect? So in this tree here, we have assembled all of the viruses that we know about from various primates, various different groups of monkeys, and also some examples of HIV that is present in individuals. And when we look at the sequence and compare it with that of the version that is present in primates, what we find is that HIV didn't just cross over into humans once, it's happened numerous times. HIV-2 is a version that we would have got from the smaller monkeys, things like the macaques, for example. HIV-2 doesn't really represent most of the viruses that we find today. HIV-1 is the one that's a big one. That's a big problem for us. And that has come over from chimpanzees. By looking to see what the closest relatives are, that's how we figure where the transmission points have come from. So HIV-1 is split into three groups, M, N, and O. And it is the M group that has done remarkably well and is responsible for the majority of the cases for HIV today. To date, HIV has infected over 75 million, I believe, which is a huge number of people. And it has also killed in excess of 30 million people as well. So understanding it is really in our best interests. How did it make this jump so successfully? Why is it this one particular group that has done so well, whereas the other ones just basically seem to fizzle out after making the jump? So one of the first questions to arise was, where did HIV actually make this jump? Where did it actually get into humans? We know that it came from chimpanzees by looking at the sequences, but which chimpanzees did it come from? So one study set out to answer these questions. We know that HIV was first described in West Africa. So they looked in chimps that are present in the West African region. And what they found was that the chimps that are present in the various regions can be distinguished by their DNA. So this tree on the left is based on the mitochondrial DNA, how similar these things are. And in this way, we can see that all of the chimps that come from a specific region generally tend to group together. Now, when we look at the version of SIV that is present in the chimps and compare it to the HIV that we see in humans, again, what we find is that the HIV that we are seeing in humans is more closely related in chimps that come from the South Cameroon region of West Africa. So this tells us that the virus originally made the jump in that area. So then the next question is, how old is HIV? When did it make this jump? And were there other things that were going on at the time that perhaps made it more successful in infecting everybody around the world? We can see that in the early 1920s, HIV remained fairly isolated. It was stuck in West Africa. It wasn't really a huge problem. I'm sorry, you can't really see the labels very well here. These basically represent areas in Africa. But around the 1950s and 60s, we can see that it started spreading a lot more rapidly. Down here at the bottom is time today, and these are basically samples that have been collected from numerous patients all over the world, and we have compared all of their DNA, or all of the DNA of the HIV, to figure out how long ago and from which common ancestor they actually derive. So clearly the spread of HIV was more successful between the 1920s and the 1960s. So what was it about this particular time period that resulted in this spread? So around this time period, Kinshasa, which is one of the major towns, cities now in Africa, was the largest and fastest growing city in that region, with transport links reaching up and down the countries. So we have Kinshasa there. The development of the railways and transport links by river carried huge numbers of workers to mining provinces all over Africa, which were reliant on immigrant labour. Records show that by the 1940s, more than 1 million people had passed through the Kinshasa rail service. So while boats and trains also spread the viruses far, other factors also played their part. Records also seem to suggest that Kinshasa had a relatively high proportion of men, and consequently there was a high demand for sex workers in the area as well, which would have increased the transmission of the disease. At the time, various health initiatives were also being administered to deliver vaccinations to individuals, but not being aware of the existence of HIV or the possibility of transmission of the disease, the use of clean needles was probably not used as accurately as it is today. These days we know about these things, so we don't do that. Another factor that might have increased its spread in areas of the more developed world would have been blood transfusions as well. So it wasn't until the 1980s that we recognized that HIV was actually the virus that was responsible for causing what we know as AIDS. It took a long time for that fact to emerge. Individuals get infected with HIV, it then takes at least 10 years before an individual is showing full-blown signs. And when individuals were coming forward with AIDS, it was a number of different symptoms. So they weren't quite sure what was going on. So that's pretty much the story of HIV. And HIV is a retrovirus, but what has that really got to do with genome evolution? So up until relatively recently, we didn't think that this particular group of retroviruses that HIV belongs to integrates into the genome. But the genomes of these two particular lemurs, the fat-tailed dwarf lemur and the grey mouse lemur, who are endemic to Madagascar, revealed that lentiviruses, the family of viruses that HIV belongs to, have actually been infecting primate genomes for a very long time. So these particular lemurs colonized Madagascar 75 million years ago. The last bit of land that joined Madagascar to the mainland disappeared about 14 million years ago. So there is no chance that these lemurs have been infected relatively recently from their primate cousins who are present in Africa carrying SIV. This is something that has had to have happened at the very least 14 million years ago. So with all of these viruses that are floating around that have been invading our genomes, you would have thought that we would have come out with some kind of defense mechanism that can deal with this. Now, we have, slightly. So inserting into the genome is not the only way that a virus can influence genome evolution. They have also influenced, to some extent, our genes. So some genes that work to combat these genomic invasions are called restriction factors. They block viral replication and are an important component of the innate immune response in viral infection. So what we have is our host gene, which encodes a restriction factor, and we have our virus. And our host gene does a very good job of blocking that virus. So what the virus does is it evolves. It evolves so that particular block doesn't change. And in response, the host also has to evolve to combat the new viral threat. Again, the virus will evolve. Again, the host will evolve. And we have this constant evolutionary arms race that goes on between these families of genes with viruses. So, HIV, does that have any restriction factors? If we look in the genome, what we find is that there are actually 3 genes that are associated with restricting HIV. You'll remember that I mentioned before that the old world monkeys are asymptomatic. They don't seem to suffer. HIV doesn't develop into full-blown AIDS in them, as it does with us. And what research has found is that when the virus enters the cell, I'm sorry, this diagram has not come out as well as I'd liked. These 3 genes, TRIM5 alpha and tetherin and APOBEC, actually disrupt the viral life cycle at different stages. TRIM5 alpha will cause the virus to uncoat, lose its protein coat very early on, which means that it can't be delivered to the correct area to do all its replication. Tetherin prevents the viral particles from actually escaping from the cell. It just forces them to clump onto the side of the cell. And APOBEC will basically screw up how the virus assembles, sorry. So why don't our genes work in this respect to HIV? Well, when we look at the versions of these genes that are present in the old world monkeys, we can see that there has been millions of years of evolution that has gone into fine-tuning these genes so that they can deal with the SIV threat. But the versions that are present in us are not ready. They weren't selected for that particular virus. So in this way, when we take HIV-1 and we infect macaque cells with it, macaques are one of the old world monkeys, we find that TRIM5 alpha does a very effective job of preventing viral infection of HIV-1. Our version of TRIM5 alpha, not so good against these things. The versions that we have in our genes would have been older versions that have dealt with older viral threats, threats that have occurred in the past. We have not yet adapted to deal with this one. Now, you might remember this figure from very early on, which shows what the different components of the human genome are. And I mentioned that this particular 8% here is retroviruses. But these areas here, signs and lines, short interspersed nuclear elements and long interspersed nuclear elements, are known as retrotransposons. Now, retrotransposons are thought to be the remnants of ancient viruses that infected early animals and inserted their genes into the genome many million years ago. These days, they can only replicate themselves by jumping about throughout a genome. They can't actually leave the cell, though. In that respect, these things are called jumping genes. Now, depending on where a new insertion might insert into the genome, it could result in disruption of a gene that we actually need, one of these 2% of genes. So it really is in the genome's best interests not to allow this to happen. So how does it do this? Given that half of its genome, roughly, well, 33% is made up of these things, how does it actually put a stop on these things jumping about? So there are a large family of proteins called the zinc finger proteins. These are DNA-binding proteins that repress the activity of these particular elements. The human genome has over 400 of these genes, and 170 of them we acquired after primates diverged from the other mammals. Now, this tells us something about the role that signs and lines have had in our genome compared to the genomes of other mammals, that we've had to make 170 new genes to deal with this threat. So how do we know that these genes actually do stop these things from jumping about? The most recently active transposons are two called SVA and L1. They're present in humans, and taking two of these specific zinc finger genes, researchers have tested this by inserting it into a mouse cell and seeing what happens. You insert a chromosome that contains these genes into a mouse cell, you infect it with these particular retrotransposons, and what they found was that the zinc finger proteins stop the transposons from jumping about. If you infect the mouse cell with just the transposons still jump about. So it's definitely these genes that are stopping these things from happening. Now, up until now, all of the viruses that I've focused on have been the retroviruses, which is indicated by the red arrow here. But with the number of whole genomes that we are getting from various species, one thing that is becoming apparent is that we also have insertions from other members of the viral families. So these stars here represent which families have managed to insert into the genomes. We're not quite sure why. In some cases, it's not so obvious. With a retrovirus, we can say that its method of replication requires that it inserts into the genome. So we can easily see why this would happen. But for example, for a DNA virus, we have no reason to think that it ever needs to insert into a genome. So why would it do that? These lines basically split this table up into the main groups of families, and we can see that there are representatives from every family within the genome of some species. They're present all over the place. So far I've been speaking about the evolutionary insight and the medical impact of what we've been finding out. But we can also use viruses in different ways for more practical uses. Some of the more obvious ones that spring to mind are the production of vaccines. We can also use these retroviruses for functions such as gene delivery. We know that a retrovirus can integrate into a genome. So let's say that we have an individual that has a non-functioning copy of something. We can insert a gene into a retrovirus and get it to deliver it to a specific area. And then there is also, interestingly, the use of viruses as biocontrol. So the production of vaccines. I'm sure that many of you are familiar with vaccines and the fact that we use viruses to make them. So the majority, well, perhaps not the majority, a large number of vaccines are made from live attenuated vaccines. And the way that this is done is by taking the virus in question, it's isolated from whoever it's infected, and it is then grown up in cells from a different species. And the reason we grow it up in cells from a different species is so that the virus adapts and gets good at growing in the cells from a different species. Then, when we take that particular virus that has adapted and put it back into human cells, it's no longer able to replicate as well in human cells, but it still causes the immune response that allows us to have immunity from this vaccine. Now, examples of vaccines that are live and attenuated would be things like the MMR or the chickenpox virus vaccine. I'm guessing that most people have heard of those ones. Excellent, some nodding heads. Now, there are other types of vaccines. So on occasion, they might decide that it's too dangerous to use a live attenuated version of the vaccine, and the viral particle might actually be inactivated by either heat or chemicals. In this way, it can't replicate. You inject it into an individual, it still elicits an immune response, and an individual still gets immunity. The same goes for the subunit vaccine, where you're just using a part of the virus, and that part of the virus might be enough just to elicit that immune response to give an individual immunity against whatever viral threat we're dealing with at the time. Generally, though, the live attenuated vaccine tends to give better immunity than the inactivated or the subunit ones. So retroviruses have also been used for a long time in molecular biology experiments to introduce pieces of DNA of interest into cells to see what they do. We call this using them as retroviral vectors, but they can also be used hypothetically for gene delivery. So in this process, cells are removed from the patient and the virus is altered, usually having its middle section knocked out so that it can't replicate, and the gene is inserted into the viral particle. This altered virus is then introduced to the individual, and theoretically what should happen is that the cells from the patient should become genetically altered, carrying the new functioning version of whatever gene or whatever piece of DNA we have introduced into an individual. Now, as far as I'm aware, they haven't managed to perfect this technique yet, so it's still a case of kind of watch this space, but theoretically it should be possible to perform this and repair non-functional copies of genes that might occur in an individual. Viruses have also started to be used as biocontrol mechanisms as well. In a lot of countries, we have problems with invasive species. So this guy here is the rabbit. I'm sure most of you know what a rabbit looks like. But in Australia, rabbits are not actually native. They were introduced there in the late 1800s. The British had gone over there and thought, what this place needs is a few rabbits, so let's take some over there. And by 1950, the number of rabbits had increased to in excess of 1 billion. It started messing with the ecosystems, it disrupted food chains, and generally caused quite a problem. Hunting them was not good enough to be able to kill enough rabbits so that they could get everything back under control. So one way of dealing with this was to introduce two viruses, the myxoma virus and the rabbit hemorrhagic disease virus. This rabbit down here is an example of one that's been infected with the myxoma virus. It usually incubates for about 2 weeks before it kills the rabbit, which gives the rabbit enough time to return to the warren and infect other rabbits as well. And it's a pretty effective way of keeping the numbers of rabbits under control. Another use of viruses would be in agriculture as well. There are many crop pests, caterpillars in particular, often decimate entire crops, and there is a virus to deal with them as well. These are baculoviruses, which are host-specific. They only target caterpillars. And what happens is that they cause the caterpillar to produce large numbers of the virus. And then for some reason, the caterpillar feels the need to climb to the top of the plant, where it hangs like this. Its body becomes swollen from the virus and eventually disintegrates, releasing the viral particles onto the plant for the next caterpillars to ingest. So I hope that my talk today has shown you that viruses have had a significant impact on genome evolution. And sometimes, it's a good thing in terms of providing us with genes and immunity. And studying genomes can tell us about viruses that have existed in the past, not just in terms of their insertions, but also by looking at other genes that are involved in tackling viral infections. It can give us a clue about the kinds of viruses that have been around in the past. That, in turn, also gives us insight into tackling viral threats that are around today. So all that remains for me to do is thank you for listening.
Does anyone have any questions at all? I didn't write anything down, so I don't remember the names. But you know the slide where you were saying that we actually have the defenses to defeat HIV, but it's not ready yet. So does that mean maybe, like, I don't know, maybe a million years in the future or something, it'll probably mutate and then we'll be able to defend ourselves? Or
What, the genes, you mean? So for a gene to change like this, what we would have to do is let evolution take its course. Yeah? The gene has to be selected, it has to increase in frequency. If a new version of the gene comes out, yeah, and it means that an individual can survive, then that gene would increase in frequency in a population until eventually everybody has it. Yeah? But that would mean that everybody today that is infected with HIV, they would not be able to survive. Yeah? So rather than waiting for genome evolution, which takes millions of years, because our viruses can evolve very, very quickly. Their genomes are quite small. With us, it takes billions of years before these things can come about. But to tackle these kinds of threats, we would be better off focusing our efforts on some kind of vaccine, rather than waiting for the genome to evolve. Theoretically, it could happen, but I don't really want to wait a million years to find out if we can deal with HIV on our own back.
Thank you.
Thank you.
Thank you. Xenotransplantation, the idea of using, say, baboon hearts in humans, has, I understand, been banned because of the worry of viruses escaping from these. Are these retroviruses that are integrated in the baboon genome?
Yes. So I believe with the xenotransplantations, it was mainly pigs, wasn't it? And there is actually an active retrovirus in pigs called PT-ERV. Which they were concerned that would risk a cross-species transmission. And in pigs, it's harmless, in the same way that SIV was harmless in the old world monkeys and the older primates. But if it makes that jump into humans, then we don't know quite what it's going to do. And yeah, we don't know about recombination occurring with the versions of endogenous retroviruses that are in the genome as well. So yes, that was a concern with xenotransplantation.
Any other questions?
Thank you very much for your talk. It was fantastic. I was really interested by the viral genes that have then been co-opted by the vertebrates. I was wondering if there are any other really neat examples of that.
Not that have been so well described. So it's not just the genes that we co-opt, but it's also the promoter regions as well that are present in the LTRs. So there are numerous instances of those having been co-opted to regulate our own genes, and perhaps, you know, it works as a better promoter that starts the gene running to do its thing than what was originally there in the genome. So in that way, I didn't say too much about that because then I've got to explain what promoters are in that. But yeah, there are numerous other examples, but none that have been so well characterized in this way and found across, you know, such a huge, diverse number of mammals.
Hi, is anyone doing any research into gene therapy of restriction factors from other organisms to be used in humans? Or is that still a bit controversial?
I'm not aware of anybody doing that kind of thing. I mean, one of the problems with the gene therapy as well is that what I didn't mention was with retroviruses, we can't specifically target them at the moment. It's not like they recognize a stretch of DNA and they're only going to insert in that place. So the problem is that you risk a retrovirus inserting anywhere in the genome, and it might be somewhere that disrupts something else. I mean, theoretically, it's possible that we can do this, but until we can target it exactly, that seems to be the stumbling block at the moment. But I haven't actually heard of anybody doing that yet about the restriction factors, or even whether it would work. I mean, to create the proteins can sometimes, that actually do this blocking effect, can sometimes be the effect of numerous genes. So it might be that you need a combination of genes, which would then get too complicated to do that kind of thing. But it's an interesting idea.
You said that it was quite obvious why viruses which used RNA had put their genes into our cells. But why do viruses which use DNA? How have they managed to do it if they don't incorporate their genes into our cells anyway?
They don't actually have to integrate into our genome. So if we go back to this slide. So the essential part here is the integration of the viral DNA into our genome, which is the green stuff. Yeah. Now, with the other viruses, they need to gain entry into the cell, but they don't actually have to integrate their DNA into the host genome to be able to reproduce. That's why it's not kind of obvious why we would expect to find their bits of DNA in our genome. Whereas with retroviruses, they have to do this integration into the host DNA to be able to reproduce. Does that clarify things?
So why do we find this in our DNA when you wouldn't expect it to be there?
It's a good question. We're still working on that one. Some of those viruses have only been found as recently as this year, in fact. You know, so yeah, we're still working on. We don't know it all yet. There's still a lot of questions to answer.
I was just wondering, with the myxoma virus, was that sort of uniquely developed, or was there something similar in other species and they thought that it would be a good idea to transport it across?
It was present. It was present, but in its early form, it probably wasn't as severe as the version that was released to deal with native species. So, as I believe, what they did was they took the version that was present already in naturally occurring populations, and they selected and they made the virus worse so that it would have this effect on decimating the rabbit population and keeping it under control in terms of numbers. But it's not something that they created from scratch. You know, that would be pretty difficult to do. It's basically building on something that was already there to make it a little bit worse, a little bit more effective in that way.
Hello. Can I first thank you for a very clear talk that even for a non-biologist like myself, I was able to follow most of it. But can I ask you, just looking at HIV, I'm aware that its background can be traced to the 1920s in Kinshasa, and perhaps the railways were the vector that brought it south, east and west, because the Belgians had developed, quite a good transportation system. And I think from postulation, from what I read in the past, that it's postulated it can be traced back to a single hunter, perhaps coming in contact with the blood of a simian monkey that he had perhaps shot or something. And I want to try to compare that with Ebola virus. I'm aware that Ebola virus outbreaks have occurred in the past and have died out. And all of a sudden now, in 2014, the world is on perhaps the edge of a massive viral outbreak without really clear what's happening. And I wonder, can you explain, has Ebola been traced back in the same way as HIV has, its method of infection and transmission? And why is this latest outbreak in the 21st century so bad compared to previous outbreaks that have died out when there were no medical precautions at all?
It's a very good question. So comparing Ebola virus with HIV, it's a little bit like comparing apples and oranges, really. I mean, with HIV, some of the conditions that led to its pandemic status also include the fact that for a long time it just went undetected. When people present with AIDS, it's a very different set of symptoms each time. Yeah, it's the immune system gone down, and then it's the secondary infection that tends to kill the individuals. Yeah, they didn't know about the virus. It also has an incubation time, though, of about 10 years before you get full-blown AIDS. Whereas with the Ebola virus, though, it's very, very quickly. I believe it's about 5 or 6 days, isn't it, that individuals start showing symptoms after infection. So is Ebola going to spread the same way as HIV? It's unlikely. I mean, it still requires fluid transfer. And because people are aware of it so much more, it's not going to be transmitted unknowingly in this way. So why has this particular outbreak spread a little bit further than previous outbreaks? So as I understand it, I don't think wiping out Ebola is something that we can achieve. It has a natural reservoir of viruses in other species, for example, bats. So individuals keep getting reinfected at certain times, a little outbreak flares up, and then it gets under control, and we've dealt with it quite effectively. In this case, with this particular outbreak, you're right, it's a little bit bigger. But is it going to get out of proportion? Is it going to get out of hand like HIV has in that way? I don't think it will. I mean, now that we're aware of it, there's been a lot of speculation on social media, on the news, is it going to go airborne? Is it going to do this? Is it going to do that? Is it going to mutate into something else? It's unlikely. It's unlikely. I mean, viruses are quite specific in terms of the cells that they target. For something like Ebola, which requires fluid transfer to go airborne, it's got to adapt to actually attack those cells which receive air, yeah? Which is a big jump. It's a fairly big jump. It's not impossible. It's theoretically possible. I mean, anything is possible. Is it likely to happen, though? Not quite. There would have to be a huge selection pressure for it to do that. Does that answer your question?
I think so, thank you.
We have time for one more question, if we have one. No? Okay, I'd just like to say thank you again to Dr Ravinder Kanda, and thank you all for coming this evening.
Thank you very much.